Good afternoon. I'd like to take some time today to discuss the innate defense regulators, and particularly desquitide, our lead clinical candidate, in terms of both their mechanism of action and their potential therapeutic applications. Before we begin today, let's start with the precautionary forward-looking statement, as Sologenics is, of course, a public company. Before we begin discussing exactly how innate defense regulators work and, more broadly, how modulating the innate immune system may lead to significant therapeutic applications, it's worth remembering what distinguishes the innate immune system from our better understood adaptive immune system. So for contrast, adaptive immunity is what we've always thought about when we think about immunity. That is, it's the antibodies and the T cells. Innate immunity is a more ancient system, in some ways a simpler system, but no less sophisticated. The innate immune system is responsible for the first response to any infection or tissue damage, and this response is both rapid and nonspecific. The activity of the innate immune system is really dictated by pre-programmed responses which are inherited through the use of specific receptors. Thus, we've heard about things like the TLR or toll-like receptors over the years, and these are really the quintessential innate immune receptors. Because these receptors are pre-programmed, they're able to respond rapidly with no learning component necessarily required. Interestingly, though, we've always thought of the innate immune system in terms of the circulating cells, the macrophages, the monocytes, and the neutrophils. However, there's a significant component of the tissue-resident cells which we're only beginning to more recently understand. And thus, really, the innate immune response is the response of the entire organism. Early studies on the innate immune system were hampered by the belief that any time innate immunity was turned on, inflammation was turned on, and as such, although deemed to be a necessary evil, the innate immune system was thought to be too primitive to really manipulate successfully from a therapeutic perspective. However, over time, we've begun to understand that, in fact, the inflammation that's dictated by the innate immune system is actually a separable construct from the tissue healing and bacterial clearance mechanisms. This was first demonstrated at the level of epigenetic mechanisms and has more recently been shown at the level of intracellular signaling pathways. Our innate defense regulators then start to leverage this difference in the intracellular signaling pathways between the inflammation and the tissue healing and anti-infective pathways. What makes the innate defense regulators unique is the way they attempt to modulate the innate immune system. Previous attempts to interact with the innate immune system have really focused on either the inputs or the outputs to the system. That is, we either tried to mimic or block these pre-programmed receptors, or we tried to mimic or block the outcome, the cytokines and chemokines that signal. However, the real control nodes for the innate immune system exist on the intracellular level. And interestingly enough, although there is a plethora of innate immune receptors that can respond to different pre-programmed signals within the innate immune system, most of these pathways actually converge at nodal points in the intracellular signaling network, meaning that there are areas where modifications of the signaling pathways can actually have significant downstream consequences. And this is precisely the response that the innate defense regulators are seeking to define. So the innate defense regulators, such as desquitide, also known as SGX-94, actually bind directly to a protein known as P62, which then alters these downstream signaling pathways such that we get more selective effector cell recruitment that enhances pathogen killing and clearance and also enhances wound healing, while controlling but not eliminating inflammation. This mechanism of the IDRs is shown in more detail here on slide 5. Here we can see that desquitide, again also known as SGX-94, is not only binding to the P62 protein, but is actually binding to the ZZ domain of this protein, which then enhances protein-protein complexes that are formed with P62 and favors specific signaling pathways that are implicated in both infection healing and tissue healing. So specifically, the interaction between P62 and the RIP1 kinase results in enhanced P38 phosphorylation and turns on specific transcription factors such as CBP beta. Importantly, though, because of the selectivity of where these IDRs interact with the P62 protein, they do not impact binding between the PB1 domain and PKC zeta, and therefore do not impact NF-kappa B. NF-kappa B, of course, is a transcription factor which is inherently associated with inflammation, and while we don't turn this off, we also don't turn it on, therefore shifting the balance between the anti-infective and tissue healing aspects of the innate immune system and the inflammatory aspects of the innate immune system. The end goal of this modulation is a change in the chemokine and cytokine production, as well as altered protein expression in various cell types, and increased macrophage recruitment to the site of infection and damage. The mechanism of action of the innate defense regulators then leads to a very broad spectrum of potential therapeutic activities. Clearly, there's an anti-infective activity to be observed, and extensive animal studies have been done in this regard, demonstrating that treatment with the IDRs both improves survival and enhances bacterial clearance. Moreover, because we're really modulating the host response and not targeting the pathogen directly, IDRs are effective irrespective of what the bacterial infection is. So it could be a gram-negative bacteria or a gram-positive one. It could be an extracellular one or an intracellular one. And perhaps most importantly, it can be either an antibiotic-sensitive or an antibiotic-resistant strain. In any of these cases, the IDRs have the same effect, enhancing the host response to the infection and bolstering survival and bacterial clearance. Moreover, the innate immune system is effective at various anatomical locations, and particularly at locations between the host and the outside world. So think of things like the skin and the lung, for example. And because the IDRs have an inherently different mechanism of action from antibiotics, they can enhance antibiotic action when antibiotics alone are suboptimal. In addition to this anti-infective activity, there's also a tissue healing activity which aids in the resolution of tissue damage, and it's actually this activity which we are focusing on the most in our first clinical indication, which we'll discuss in more detail later in this presentation. And finally, the IDRs also modulate inflammation, and they modulate it in a specific way, shifting the inflammatory state to a more anti-inflammatory response without completely ablating the inflammation itself. This is important because, as we all know, inflammation has a critical role to play. It's just that it's often an over-exuberant response. And finally, more recent research has certainly suggested that, much like when we modulate the adaptive immune system, modulating the innate immune system may also have a significant impact in the context of oncology. The innate defense regulators have been extensively studied in non-clinical animal models, and I'm going to show a variety of different aspects of modulating the innate immune system to therapeutic effect in these animal models. As I've mentioned, however, there's been significant progress made with our lead clinical candidate in our first clinical indication, and we'll also be discussing this towards the end of the presentation. So from the anti-infective perspective, early attempts to modulate the innate immune system, which really focused on modulating the receptor binding to specific agonists, such as the TLR agonist, showed that prophylactic administration of innate immune regulators could certainly aid in the prevention of infection. However, these compounds, because they turned on the innate immune system in all its aspects, including inflammation, were not as successful in the therapeutic setting. Here we show that the innate defense regulators, because they have an inherently different mechanism targeting that intracellular nodal point in the signaling pathways, actually has a dramatically different profile. While there is definitely prophylactic activity, as we've demonstrated in a number of our publications, there is also, importantly, therapeutic activity. So here we see two examples. On the left-hand side, improving survival in animals infected with a gram-positive antibiotic-resistant bacteremia, specifically methicillin-resistant Staph aureus, with a statistically significant improvement in survival with the treatment with SGX-94 or desquitide. And on the right-hand side, a different survival study, this time with a gram-negative leukopenic septicemia, specifically caused by Pseudomonas aeruginosa. So the interesting thing here is, again, that because we are modulating the innate immune response and not directly targeting the bacteria, we have very broad spectrum activity. And in both cases, the therapeutic administration occurred a significant time after the initial infection, and when either frank symptoms appeared on the right-hand side or when mortality had begun to become inevitable, as shown on the left-hand side. Of course, one of the most obvious questions about the innate defense regulators is how they mediate the responses that are seen when they bind at these intracellular convergence points in the innate immune signaling pathway. And we find that the ultimate mediators of the innate immune response are actually the macrophages. So we find that we always get increased macrophage recruitment to the site of infection or tissue damage, as is shown here. Importantly, we don't get a concomitant increase in neutrophils, and this can be quite important because the neutrophils are actually associated with a more inflammatory response. It is important to note that infection targeting is an intrinsic response of the innate immune system. It isn't necessary for us to target the delivery of the innate defense regulators. The targeting to the response of the infection or the tissue damage is inherently carried out by the innate immune system, and we simply bolster that response. It's also important to note that because the effect is mediated by the recruitment of these cells, we have actually an enduring pharmacodynamic effect despite a very rapid pharmacokinetic response. So while the drug can be processed very quickly, its administration, when it surpasses a certain threshold levels, sets up a change in state in the innate immune system which lasts for a very long time. The other advantage of this approach is that because the response lasts for a long time, even when the drug is not present, there are no drug-drug interactions to be concerned about in the context of a polypharmacy setting, such as infection or oncology. Now, I've mentioned earlier that the innate immune system is also, of course, intrinsically linked to inflammation, and that one of the roles of the innate defense regulators is to modulate the inflammation without completely shutting it down. Here we see two examples of this broad spectrum anti-inflammatory response in two different markers. So on the left-hand side, we see that there's been an infection in the peritoneal cavity of a mouse, which has driven inflammation in the case where the infection is untreated. Here we see on the left-hand side the saline group. Whereas in the context of treatment with the SGX-94, we can see that by 24 hours post-infection, there's been a statistically significant reduction in the amount of TNF-alpha being secreted in that peritoneal cavity. Another way of looking at this is shown in the graph on the right-hand side. Here we're looking at a lung inflammation model where the lungs of the mice are induced into an inflammatory state with the use of endotoxin. And here we see that an anti-inflammatory marker known as IL-1RA is actually enhanced with treatment with SGX-94 or Descretide. And this really represents the type of inflammatory control that the innate defense regulators provide. So they don't completely ablate any one response, such as completely ablating TNF, which comes with its own consequences, as we've seen with some of the anti-TNF drugs on the market, but rather we get a modulation of the TNF response, but also a modulation of other responses, such as IL-1 alpha and beta, and anti-inflammatory markers like IL-1RA and TNFR2. So it is a change in state of the entire innate immune system, as opposed to a singular response in a single unidimensional response factor. So I mentioned earlier that the response to the innate immune system is driven not just by infection, but also by tissue damage. And this is seen here on a model that was done in mice where their skin was deliberately damaged in sort of a minor way, but then infected with methicillin-resistant Staph aureus. We can see on the left-hand side that both the antibiotic linezolid and SGX-94 or Descretide have significantly reduced the amount of ulceration that's observed relative to the untreated control animals who receive saline. However, what's intriguing is that not only is the infection reduced, but in the SGX94 group, also the swelling score is reduced at 96 hours post-infection. The swelling score is associated with the healing of the tissue, and this appears to indicate that tissue healing is enhanced in the presence of the innate defense regulators, unlike what is caused by the antibiotic alone. Following up on this tissue healing activity, additional studies have been done in the context of oral mucositis. So oral mucositis is really a response of the innate immune system to the damage done by radiation or chemotherapy, particularly in the oral mucosa, but also throughout the gastrointestinal tract. Of course, the damage done by the radiation or chemotherapy is inevitable, but interestingly, it is actually the innate immune response to this damage, including the inflammatory component, which actually further exacerbates the degree of damage that is seen. This ultimately causes ulceration, which can be a portal of entry for infection, and then ultimately, of course, there has to be tissue healing. Now, clearly, oral mucositis is a very significant clinical condition, one of the most debilitating side effects of cancer chemotherapy and radiation, and something that has been known to limit the ability of patients to fully pursue their anti-tumor treatment regimen. Any attempts to modulate oral mucositis clearly must not interfere with the actual tumor-directed radiation and chemotherapy response, and therefore responses that are focused on the innate immune system appear to be optimal. We initially evaluated this in the context of two different animal models. On the left-hand side, we have a radiation-induced mucositis, and we can see there's been a 50% reduction in the duration of oral mucositis that was observed in these animals. And on the right-hand side, we have a chemotherapy-induced mucositis. Now, while the radiation-induced mucositis was clearly occurred where the radiation was directed, in this case the cheek of the animal, the chemotherapy-induced mucositis is a more systemic insult, of course, and here we see both the 50% reduction in the oral mucositis, but also a reduction in the degree of colitis that's observed in the lower gastrointestinal tract as well, indicating again that the innate defense regulators change the innate immune response at a systemic level, which then allows the innate immune system to more appropriately target the damage that has occurred. Following up on this concept of the inflammatory response, another gastrointestinal injury model has also been performed, which models the effect of inflammatory bowel disease. In this case, an oral dextran sodium sulfate solution is given on days 0 to 5 to rodents, which inevitably damages the GI lining of the lining of the GI tract. When we administer SGX94 or dusquatide, either before, during, or after the insult, in all cases we find that we're able to modulate the severity of colitis in that GI tract in response to that insult. And these responses are qualitatively similar to those that are achieved with steroids. However, in the steroid case, the animals are dose limited because of very rapid weight loss, whereas that is not the case with the treatment with dusquatide. Now, I've mentioned a couple times that there could be a role for the innate defense system in oncology, much like there's a role for adaptive immunity. And here we think of things like the checkpoint inhibitors or the CAR-T response. The role of the innate immune system could be quite a bit broader and could be thought of in two ways. One, a direct effect on the cells themselves, since clearly they are mutated human cells, which also have some capacity to respond with the same innate immune signaling pathways, as well as a response in the tumor microenvironment itself. Specifically with the innate defense regulators, this can actually be thought of in these same two ways. Firstly, the p62 protein which the innate defense regulators target is also expressed in tumor cells, and the p62 protein is actually enhanced in chemotherapy and radiation treatment resistance, suggesting that there may be some utility to targeting the p62 response directly in tumor cells. Secondly, of course, the innate immune system is a key component of the tumor microenvironment, and it has already been demonstrated in the literature that P62 expression in stromal cells really impacts stromal cell support of multiple myeloma growth. Both of these are just preliminary examples of how we might consider pursuing oncology, not just from the point of view of T cells and antibodies, but also from the point of view of innate immunity. Following up on these theoretical thoughts are some very early preliminary studies we've conducted. On the left-hand side, the idea of directly targeting the tumor cell itself. We have an MCF7 xenograft model here where the MCF7 cells, which is a breast cancer cell line, are implanted into the backs of mice. And we see that the administration of SGX-94 not only doesn't support growth of the tumor, which is very important when you think about it from the context of a clinical indication such as oral mucositis, but actually shows a potentially significant effect on reducing the tumor growth through time. In the right-hand side, we have a very different model. In this case, what we've done is we've taken some stromal cells and pre-incubated them with SGX-94, and then asked how well those stromal cells help the multiple myeloma cells to grow. Here we can see that relative to cells that have been incubated with saline, when the stromal cells have been pre-treated with SGX-94, they are less able to support tumor cell growth. These early results have been published, but clearly our initial indication is really in the supportive care aspect of oral mucositis. Nonetheless, this may present a growth potential for modulation of the innate immune system and the innate defense regulators themselves in the longer term. Of course, one of the most important questions that has to be asked whenever there's a potential therapeutic modality is to question how well it's really going to translate to the clinic. Of course, there are many things that we can treat in small furry animals that somehow do not translate to the clinical setting. Because the innate immune system is present in all orders of mammals and very highly conserved, it was believed from the outset that there would be very good translation from the rodent and other animal models into the human clinical scenario. The target protein P62, which the innate defense regulators interact with, is very highly conserved, with 91% sequence identity between mouse and human and 99% between orangutan and human. To date, we've completed a phase 1 study in 84 healthy volunteers, which I'll show you some data for, and where the drug was very well tolerated, as well as a phase 2 study in 111 head and neck cancer patients at risk of severe oral mucositis, where we demonstrated a significant reduction in the median duration of severe oral mucositis. To date, we've seen a complete concordance between our non-clinical and clinical findings, and we are now actively enrolling a phase 3 study in about 190 head and neck cancer patients at risk of developing severe oral mucositis. Our phase 1 study was our first opportunity to really evaluate the potential anti-inflammatory action of the innate defense regulators, as well as obviously assessing their safety and tolerability. The administration of dusquatide or SGX-94 was not associated with any serious or severe adverse events in the course of the phase 1 study, which evaluated single doses as high as 8 mg per kg and 7 daily repeating doses as high as 6.5 mg per kg. No maximum tolerated dose was identified, and all drug levels were well tolerated. However, one of the other things we were able to do in this study was to collect whole blood samples from the volunteers at various time points post-dosing, and then stimulate those blood samples in the test tube with endotoxin to induce an inflammatory response. Interestingly, we found that when the volunteers had been treated with lower doses of dusquatide or SGX-94, we actually found a stronger anti-inflammatory profile. And that profile was observed both in the context of increased anti-inflammatory markers as well as decreased inflammatory markers. Thus, you see significant effects, for example, in the ratio of anti-inflammatory IL-1RA to inflammatory IL-1 beta and anti-inflammatory TNFR2 to inflammatory TNF-alpha. Moreover, this response was not only seen at 1 hour post-dose, but could be sustained, with responses also observed as late as 96 hours post-dose. Again, reflecting that enduring pharmacodynamic effect that had been previously evaluated in the non-clinical finding. And in fact, this dose-response profile, where we saw more anti-inflammatory responses at the lower dose level, was actually also seen in early non-clinical studies, especially those conducted in primates, where a larger range of dose levels could be evaluated. Importantly, clinical studies, subsequent clinical studies to this, actually evaluated both of these dose levels: the low dose level, where we went forward with a 1.5 mg per kg dose, and the high dose level, where we went forward with a 6.0 mg per kg dose. Based on these results, one would anticipate a stronger anti-inflammatory response at the lower dose level in these subsequent trials, and this is precisely what we set out to test. So our phase 2 study, as I indicated, was in the domain of oral mucositis. It was a multi-center randomized placebo-controlled study where we enrolled 111 head and neck cancer patients that were planned to receive at least 55 gray of radiation to the mouth, and either weekly or every third week cisplatin. We found, as shown on the left-hand side, that there was a 50% reduction in the median duration of severe oral mucositis in the patient population overall. And this is, of course, very consistent with our non-clinical results, where again we saw a 50% reduction in duration. Moreover, we also identified a high-risk subpopulation, in this case specifically the population receiving the higher dose levels of cisplatin, which were more susceptible to oral mucositis and experienced more prolonged oral mucositis. Again, we saw a statistically significant reduction in the median duration of severe oral mucositis in the subpopulation. Note, however, in both of these cases that the most significant effect occurred at the lower, more anti-inflammatory dose of 1.5 mg per kg, very consistent with our phase 1 studies and our non-clinical primate studies we had conducted earlier. One of the interesting aspects of this study was that, in addition to being susceptible to severe oral mucositis, patients receiving chemoradiation therapy are also, of course, susceptible to infection. And infection was recorded as a monitored adverse event throughout the course of the study. Now, in this case, all concurrent antibiotic treatments are allowed, both prophylactic and therapeutic, and the only question was whether an infection occurred, which could be both fungal and non-fungal for the most part. Here we see that there was a statistically significant reduction in the incidence rate of infection in the SGX-9/4 treated groups, both when we considered all infections and when we restricted our analysis to non-fungal or bacterial infections. And this again was very consistent with our non-clinical work that I've shown you earlier. And finally, of course, from the point of view of a cancer supportive care indication, we were also concerned, from a safety perspective at least, about any potential for accelerated tumor resolution. And so here we see that both at 1 month post-treatment and at 12 months post-treatment, there is a benefit accruing to the SGX-942 or Dusquotide treated group with an enhanced resolution of tumor. Now, while we aren't at this moment pursuing SGX-942 as a direct oncology application, this nonetheless is very comforting from the point of view of a cancer supportive care indication, where clearly we are not affecting the tumor in a deleterious way. Consistent with these results, where we saw the sort of accelerated tumor clearance, we are also seeing a minor survival benefit accruing to the SGX-942 treated group, though of course in the context of head and neck cancer, mortality over the 1 year or 12-month time frame is not very significant, thankfully. So as I mentioned earlier, we continue to develop SGX-942 in the context of oral mucositis, and we are currently enrolling in a pivotal phase 3 multinational study, again, of course, blinded placebo control. Here we're focusing on head and neck cancer patients that are receiving chemoradiation therapy, including at least 55 gray of fractionated radiation and the higher 80 to 100 mg per meter squared cisplatin every third week. The study is randomized 1-to-1 and is actively enrolling in both the US and Europe. This program has Fast Track designation, Promising Innovative Medicine designation from the UK MHRA, and the EMA has acknowledged that a single phase 3 study may be sufficient. The study is also supported by a National Institute of Health grant award. In summary then, the innate defense regulators really offer a unique therapeutic approach. They have a unique composition of matter with small molecules that are novel and not previously known. They are peptides composed of all amino acids, which means the metabolites are not an issue, and moreover that degradation is driven by proteases and peptidases, which eliminates any potential drug-drug interactions with drugs that are primarily metabolized by the kidney or liver. Moreover, they're small peptides, which means that they are very simple to synthesize, both using solid and solution phase synthesis, and they are unlikely to induce immunogenicity. These compounds have a unique mechanism of action, as I discussed earlier, targeting an intracellular convergence point in the signaling networks, and in an approach that's never been really attempted with the innate immune system before. A consequence of targeting this intracellular convergence point is that we can down-modulate acute inflammation without increasing susceptibility to infection, and that targeting to the disease site is really driven by the innate immune system, so a single mode of administration is sufficient. Phase 1 studies have indicated that the drug is very well tolerated, up to 8 mg per kg, which was the maximum dose tested, and can be rapidly administered as a convenient 4-minute infusion for a hospital-based population. There is, of course, the potential for an EpiPen-like administration using an automated delivery system, an automated injection device, if warranted. From the efficacy point of view, we've seen potential therapy where in any context where the innate immune system is triggered, so we can think about infection and tissue damage and oncology, just for starters. I hope I've convinced you then the innate immune system has a broad range of potential indications as shown on this indication wheel on slide 23. Clearly our first endeavors are really focused on the inflammatory and tissue healing response of the innate immune system with the clinical indication of oral mucositis. However, we are rapidly scaling up our efforts in areas such as emerging and antibiotic-resistant infection, and in the longer term, potentially in oncology and other associated areas. Many of the results I've presented today have been previously published and are shown here on the slide with key references should you desire to see more of the data in detail. Thank you very much for your time.